I'd love to thank the Pankaj, who's I'm extremely pleased to meet because your work I've been following for the last few years, but I don't think we met before. Abhishek I've known for a much longer time, and I would have liked it if he had joined us here at JNU in Delhi, but I'm glad that he's come back to India at least, and we'll be able to get something going off the ground. And this is a fantastic meeting. The presentations have been excellent yesterday and today, and the program also. Now, my talk is largely to go a little beyond what AI is doing, because I've done this with intent. There's a lot of use of AI in bioinformatics, and it's largely in biomarker discovery. It's moving from biomarker discovery to risk prediction, but there's a lot of traditional bioinformatics, especially in the four structures that we look at: sequence, structure-based, the systems biology perspective, and also now with big data, in being able to extend. Essentially what AI does is give you a correlation between some signal that you're measuring to a disease or to some outcome to identify causality. So let's start with simply a panel. Really, the last presentation was probably beyond excellent and will overlap much of what I'm going to say. But let's look at diagnosis in its tradition before we've come up with AI. I'm going to put them into two buckets. The first is what I'm going to call threshold-based diagnostics, and the second is a visual inspection-based diagnostics. Can you see the marker by any chance? The pointer?
Yeah, we can see the cursor, but not the highlighter.
All right. No, as long as you can see the cursor, that's fine. So, okay, let's go ahead with it. Now, see, the thing with biochemical assays, I call them threshold because traditionally you have some sort of a point beyond which, let's say 110, you have a tendency towards diabetes, blood sugar measurements. Less than that, you're okay. Those kind of biochemical assays, you've got microbiological assays. These are standard lab assays. Now, where does the role of biomarker discovery come in new and improved multivariate diagnostics with data mining? Essentially, with images, you have deep learning, and deep learning on images is traditionally has improved a lot in the last year, and it's really applied towards radiological data, and you can automate visual interpretation of these. Now, here's the thing with threshold-based diagnostics. This is a paper that was published in February 2018 in Science, which is detection and localization of surgically resectable cancers with multi-analyte blood tests. Essentially by taking, if you look down here at the abstract, 850 healthy control individuals, 1,000 patients previously diagnosed with cancer, and you simply assessed their circulating protein biomarkers and tumor-specific mutations, which are traditionally blood analytes that you would measure using these standard things. So you've got now a matrix of various two classes, the patients and the healthy controls. And this data set itself has been cited approximately about 450 times within 1 year by data scientists who improved on the prediction, which was earlier 98%, to be able to identify your blood cancer simply from a blood test using what would have been standard blood analyte measurements. So these are multivariate, meaning that you're not taking simply one, you're combining all of them together, and then you're being able to use them towards improved diagnostics. This because honestly, it's quite commonly now available and everyone's very excited with image analysis. But the bottom line is, for instance, if you have lung cancer, lung diseases, you use them for tuberculosis, you can even now use them for COVID and very quickly done. You have image analysis where you can use these deep learning methods to very quickly identify and accurately predict the outcome disease. So again, a diagnostic. More interesting for those of us working in the lab, especially with sequence, is the ability to get SNP, indel, structural variations from genome variation. Now this is with NGS become even more standard, the cost has become low, and it's rapidly becoming a common biodiagnostic. So you have disease detection. You've also got risk prediction, which I'll come up now with breast cancer, gene panel tests, etc., etc. And our aim is, again, can we use data mining and machine learning to get new dimensions towards these genetic diagnostics. Now, there are standard ones available. For instance, for breast cancer, there's a BRCA panel, a panel of about 21 genes, and you can predict breast cancer onset with a relatively high level of accuracy. Now, the other ones I'm not too sure about but they are out in the market. These are box sets where, based on the analysis of those two classes, prediction of onset of disease and the control, you have variation analysis to be able to predict the structural variations, SNPs from genome variations, to be able to predict the potential onset, and this is therefore a risk of getting cancer. Now, there are a number of other methods that are coming up. More importantly, in epigenetic variations, histone modifications, methylations, and in the single cell sequencing of transcripts, expression, differentially expressed genes, etc. And these are what we're going to use now in the future. There's a lot of data coming in, and newer cancers, colorectal cancers, glioblastomas. The last talk has really made this slide relatively redundant, because now you know that even circulating blood cells and circulating DNA, transcript DNA, are used to be able to do this. Now, the point is that the detection of relative abundance of a transcript with tissue-specific effects, you can use this for cancer diagnosis. Now, this is what I look at as the data panel that we are presently interested in. And collecting this kind of a data set is what we look at as a starting point towards applying data mining methods or machine learning to be able to improve diagnostics. This wasn't really presented, but it's one of the more interesting papers that I think of. Example, for instance, is that by collecting platelets, this is, I think, 2015, even though it's updated in 2018, you have variations in the platelet pattern at the RNA expression when you're monitoring them. And you can then identify what kind of cancer simply from the platelet pattern. So this is, the platelets are educated by the tumor to produce a different pattern, which we can then use as an early diagnostic. So this is again without any, this is simply a blood test and then sequencing and then get down. Now this is itself with clustering, because it's single cell and then you do separation and you do clustering data. These are the algorithms that we are very interested in being able to differentiate between these various classes and mark with a higher level of accuracy whether we can detect the onset of or the disease, the early onset of the disease. So you're really looking at very early cancer detection. Okay, now let's move on to the main portion of what I'm really interested in talking about, which is my own work given this kind of a data. So given that you have genetic variations, they could be SNPs, there could be indels, there are structural variations. The point is that what is the cause for the disease? This is the marker. You have a measure and you have a signal. What does it cause? Now, at one level, at the protein structural level, which people of my generation spent a lot of their youth in feeling very excited about, the structure of a protein and its can detect what, and its differences to mutation can detect disease. So there is the static structure to be able to get signals from the sequence and correlate them to that structure. And there is also the molecular dynamics and simulations. This is a free energy plot, which I'll show you in a later slide, to be able to understand the protein's function, its loss of function, and its modification of function that causes this disease. Now, I'm going to spend a little bit on this before I move on to the slide. And why is it important that when we identify the cause of the disease, we then might be able to identify the therapy. And it's not simply by trial and error that we are identifying a therapy. We can design a therapy specific to that particular problem, and we have the structure. Okay, so let's move on now. Now, the first kind of work, which was mentioned in the introduction, was done by my team, where we've used hidden Markov models about 15 years ago, but we've applied them more recently as well, in modifying it to be able to apply the hidden Markov model's patterns to detect the residue's importance for its function. I'll repeat that again. with reference to what we are doing with this. Here is a typical multiple alignment of 4 different kinds of proteins, which all have the same structure, but they will have different residues of importance, and therefore different, in this case, is a protein kinase, different substrate specificity. So the point is that, okay, their structures are all very similar. What is it that causes a difference in substrate specificity? So there are different kinds of residues. Some residues are responsible for the overall homology conservation of the protein, and they will be conserved across all proteins. Some, on the other hand, will be conserved only within that subfamily and differently conserved in other subfamilies. Now, if you have the same active site, the change of a residue over here is responsible for the change of a substrate specificity. You could also have other protein families. This, for instance, is something called the Tim barrel family, where you have the evolution of active sites or binding sites on different parts of the same fold. So these will have patterns in the sequence which are conserved. They may not be even present in the other because the overall site may not be present, but they will not be present or conserved in the other sites. So now what we want is a method that we can pick out from the probability matrix, which is a hidden Markov model, to be able to detect these kind of residues, conserved in one and not conserved in the others. And why is it important for our purpose of disease detection? If we have a mutation in this residue that is responsible for substrate recognition or responsible for the thermodynamic functioning of the protein, then there would be critical residues, and therefore those critical residues can be identified along with where they cause their function. Okay, so as part of that HMM MODI, what you do is that let me just quickly run through this particular equation. And for those of you who don't come from a mathematics background, essentially what we'll do is look at I and Q as 2 of these subfamilies and the probability of the conservation of a residue. The color coding here may not be very clear on your screen. So you have red as something that is conserved across the entire family, and you have blue as something that is conserved in one family and differently conserved in another family. Okay, so if P and Q, which is the probability or the conservation patterns of both of them, are the same, this reduces to 1, and therefore log of 1 reduces to 0. The relative entropy or the entropy calculations reduce down to 0. However, if they are different, these values of P and Q, then the relative entropy is high. So these are just basically, when you have relative entropy calculations, you can select out residues based on the entropy on whether something is going to be similar or something is going to be differently conserved. So this is traditionally used with protein sequences. It was used in our pattern recognition. But what we're doing now is to use them to identify, given a mutation in a protein, how will this protein affect, or this mutation affect, the conservation. So again, back to this particular screen, we can identify what's wrong with these residues, and therefore we can map them back to the structure. And now, not all residues are of importance to the eventual function. So the residues that are important, if they are mutated, they are responsible for the loss of function, which may be associated with the disease, or the modification of the function that may be associated with the disease. So you can identify those residues, and then you can look at how to correct them using some chemical perturbation, which is traditionally part of drug discovery, out of the scope of what we are talking about, but that's where we will move towards the next level. So if that is the static identification, or rather the identification of the residue from a static perspective on the structure, what happens is that if that it does not really affect it, but it's not in the active site, perhaps it's somewhere far away. So it's distal from the active site. Somehow you don't seem to understand, or we don't seem to understand, why a residue far from an active site should be associated with the binding. So here's a research problem, which is, for instance, the RNA binding proteins or the DNA binding proteins in neurons. TDP43 is a good candidate. Now, these are associated with clumping of these proteins, which eventually cause many of the diseases, including Alzheimer's, which cause the clumping of proteins. Now, the clumping of proteins is because of a misfolding. Now, when you map out the protein variation in the human population, this is the protein sequence shown in a sequential way with the secondary structures associated. You have variation, quite a lot of variation in the human population. And what is therefore the prediction towards this variation in these parts of the domains associated with this protein? And how does it cause function? Now, if you use molecular dynamics, also very well developed, you might be able to use, what we do is we use perturbation methods where we can heat the protein. So this is heating the protein from 300 and the yellow plot is at 500. So we are basically melting the protein, and you take the native protein and you take the mutant protein and you see whether they're, after unfolding, by studying together the conformations, whether they have a free energy surface which has buckets, which can get some sort of a substructure which is relatively stable, and that substructure may have a hydrophobic surface which therefore allows for clumping. Now, many of these are identified. This is a typical example with TTP43, which was published, I think, about 2 years ago. So essentially what you do is you identify these kind of structures. You find that some residues are outward and exposed, which cause aggregation properties. And perhaps by blocking that, that would be a more suitable way of being able to solve Alzheimer's disease. It probably needs to be done much more as a prophylactic than a therapeutic, but those are early days. The point is that you can identify the reason for clumping from the structure and from a thermodynamic perspective. Now, there are other reasons that we should be interested in proteins. This is a big system, also becoming more rapidly available towards computation now. This is a membrane protein. And it's a typical membrane protein from Mycobacterium tuberculosis involved in drug resistance. Now, this is also of importance to us. If there is a mutation in the protein and the drug is no longer being pumped out by this, how do we identify which mutations can cause the reduced resistance and be able to reverse susceptibility? So we are able to simulate the entire protein. Now, this is largely put forward as an exciting, not so much as an exciting example. This is the way that by moving the protein, we identify that a path can be formed, and then the protein's small molecule is either being pumped out or not being pumped out. That would be the drug efflux mechanism. So the efflux mechanism is understood by using the simulation. Now, the purpose in being able to say this is that in the last 5 years, even simple gaming machines with the adequate software can take on large systems such as membrane proteins, which were earlier in the realm of supercomputing spaces. So the bottom line is, given a mutation in one of these proteins, can I identify whether it will have an improved efflux, reduced efflux, or would we have other drugs? Now, this can also be used to test whether a drug in a present multi-drug resistant strain would be effective or not using a computational method. Okay, so we finished with these two, which is on the protein structure and also using protein structure dynamics. Now, I will not spend much time, because I've removed the slides, on how we implement systems-based studies. But looking at this slide, one is that you use a network, which is essentially from metabolic pathways such as the KEGG or the protein-protein interaction pathways, which are string, and we use graph-based algorithms. If there is a modification on one of them, then what is the impact on the overall graph, and how can we use perturbations about them? Now, we want, more importantly, to be able to see how we can build these graphs and then look at the difference between these graphs from the genome variation data. What are, for instance, the similarities and difference among distinct tumors? So if we go back to this kind of a data, genetic testing, which is used for risk prediction, what we have now is that, I've finished with the slide, but I'm going to repeat most of these. They are SNP, indel, structural variations. We want to identify histone modifications, methylation, transcript expression, THEs. Now, at bioinformatics, there's another problem. It's nice to be able to get everything in a nice matrix, which has just a listing of where the variations are, the indels are, the structural variations. But the raw data that is generated is normally in a factor of 1,000,000 more than what we are dealing with here. You have raw data as reads, and being able to get the structural variation information is pipelines of standard algorithms or standardized algorithms. There's constant improvement in these algorithms. And our aim in a laboratory setting or a computational biology laboratory setting is to be able to run these algorithms locally on hardware, which may not be the standard hardware. I mean, some of these data sets require much larger hardware. So being able to set up this using pipelines. One of the workflows that we use, a workflow manager, is something called Galaxy. I see that there's also going to be a talk on Galaxy for proteomics later, but it's a skill set in bioinformatics to be able to provision these tools for other people's use. So this is what we had developed for standard NGS. You have reads. In the process, you want to concatenate them. There's a normalization. In this, it's not the statistical meaning of normalization. Here, what we want to do is to be able to remove redundant reads and have only one common read, because then the algorithm can work much faster. The software Trinity, which is used for de novo data sets, for human data, it's not so important, but it's for novel metagenomic data sets, it becomes quite useful. And then you want abundance estimation for differential expression. There's another algorithm that you would use for detection of variations. And this is simply something that I'm just running through to be able to say that all these tools must be available and the data must be ported, and ported perhaps on different computers, which run specialized tools and are optimized for specialized tools, some requiring higher number of CPUs, some requiring larger amount of RAM. So we have now shifted over to using workflows. There's another one called Nextflow, but something called SnakeMake, where you can take your script and update it, and there's an explicit syntax that is used for pushing and pulling. And here, for instance, is a standard variation pipeline using BWA, SAM tools, and indexing. And we put your workflow as a shell script, and we convert it into a workflow manager using SnakeMake. Now, there are two reasons why we want to do this. The first is that the backend computation is not standard for a script. It needs to be accessed using schedulers. And schedulers need workflow management systems. So you need to break each of these shell script units and be able to send them out and retrieve the results. And that's where we need to have specific workflow management systems. Now, workflow management has been something of an interest to me because computational infrastructure management is a skill set in bioinformatics, often overlooked, but is actually a very useful skill set. Two of my PhD students, for instance, now run large facilities, one at EBI, largely because of these skills. And that's why I sort of put it forward. When we are dealing with bioinformatics, don't just look at what we can do with reference to getting results, also how we provision these to be able to get tools running. So most of my systems-based analysis, I'm focusing this particular section of my presentation on to be able to say that the data sets for systems are very, very large at the raw data level, and you need to have specific skills to be able to translate them, to be able to get the results that you're interested in. So once we get these results and we overlay them back onto, for instance, network-based modeling and get similarities and these metabolic pathways, there are standard simulation tools, graph theoretic tools, and measures. Some of them were presented, for instance, in yesterday's excellent talk on the neuroscience graph-based method to be able to detect certain aberrations in the brain structure. And you could use flux balance analysis for metabolic pathways. Okay. So now we have raw data. Managing it itself is a big issue, but we have this. And we have a lot of processed data with specific skill sets which are now becoming relatively historical. For instance, protein structure. Biophysicists, of course, will defend themselves and say it's not historical, but I'm saying that there's not really much development in the methods over here. So you have metabolic networks, transcription networks, protein structure. The signal transduction, transcription networks, intercellular are still to be done. Protein interactions and protein dynamics are standard methods that we can apply. When I say transcription networks and signal transcription networks are still developing, they will be developing, but they are cell dependent, different between different cells, and there's not complete information available, but they are coming up. The point I'm putting them together over here is that given this kind of a raw data, we will put various methods that we know from our standard understanding of sequence to structure and systems analysis using our biochemistry, bioinformatics approaches to be able to predict causality and therefore to be able to use, again, relatively standard computational methods for therapeutic interventions. Now, this is from data that is standardly used in diagnostics. And what are we looking at is a near future. This, for instance, is a point-of-care device which seems to measure non-biochemical data. For instance, ECG sensors. This is a toy really, but so you've got a spirometer, glucometer, you've got this oxygen pulse meter, you've even got a snore sensor. The bottom line is that there are, based on Raspberry Pi or Arduino kind of structures, you can detect sensors or add-on sensors and develop sensors and get a lot of measures. And this is the kind of data that you will get that cause or are associated with disease: dietary habits, activity measurements, fitness parameters. Fitness parameters is a done thing. There are tools like Fitbits and standard watch-based measures that you can put in. There are stress levels, and there are also environmental conditions that you come in. Now, this adds a different layer of data and it enables us to move towards much more than simply biochemical-based causality, towards a combination of what would be your biochemical would be your genes, and this would be a measure of your environment in being able to detect what is the impact on disease. So using data mining and machine learning and also using network analysis, we will get a relation between environmental factors, lifestyle, risk assessment, and using the interconnections and pathways we have from network analysis, we can look at what we call now a systemic cure. And this is important because diseases that are responsible for what we call rare genetic disease, for example, for instance, sickle cell anemia is a single mutation. Those are relatively easy to detect. It's the bigger problems that diabetes and complex disease that require systemic understandings and therefore systemic cures. And very often the data sets that we get, even when we have large numbers of data sets, don't give us enough information. So there is a relationship that we need to factor in between environmental factors, lifestyle, and risk assessments. Now, this is early work, meaning that it's about 4 years ago, to show you why it's important, not so much responsible over here. So this is a paper of mine from the Malaria Journal, where we were looking at socioeconomic, epidemiological, and geographical features that we overlaid onto a map to identify malaria hotspots. Now, the traditional use of malaria hotspot identification is to predict mosquitoes and monsoons, and there is a correlation, but that's not a hotspot. That is generally at a very high level, we'll say that malaria is onset. The hotspot that we're trying to identify is, again, back to causality. It's not the measurement after, it is the prediction. Given these conditions, can we predict that malaria is going to come up in these areas? Now, this is the data that you have. You have socioeconomic aspects, population, child population, literacy, workforce participation. One of the advantages of JNU is that we host the census data. So this kind of data can be overlaid onto a map. There's epidemiological data. In this is the actual measurements of the onset of disease. such as slides collected, examined. And then you have geographical features. Some of them are settlement, forest covers, water bodies, rainfall, relative humidity, and temperature. The correlation between epidemiology and geographical features was very, very high. You can easily detect between water bodies. So essentially, you can find out areas which have a potential for hotspot. Now, this has a potential for getting malaria, but it doesn't always overlay. There are areas, and it's not even poverty, it's not literacy that cause. In many cases, most of these don't correlate with the onset or the prediction of malaria. The one that does, surprisingly, is workforce participation. Now, this is understood if you talk to an epidemiologist. If you do not have much of your local population involved in construction activity, you are importing migrant labor for construction activity. Now, construction locations have a very high degree of bad water management. And these are the areas which cause malaria hotspots. Because migrant labor brings in malaria, and migrant labor applied to construction activity causes hotspot prediction. So a lot of these that we're looking at, where you talk about natural forest cover, mosquitoes, etc., these are small vectors. And the real reason that was happening is that you have migrant labor coming in with the disease, and then they are spread after they come up with this. This example is merely to show you that sometimes, very often, the data set that you're looking at that is actually causative is not really something that you would think of right at the beginning. My present work is to, this is from the News and Views of Nature last year, is to mine research papers where we've collected about 90 million scientific research papers. This is an area in AI, which is text mining. And we're picking up various patterns to associate, again, the short term will be to generate data sets associated with life sciences research and biomedical research to be able to apply them towards understanding what we can get out. Because nobody can really read research papers, especially when you don't have access to them. Okay, so this is my final slide, I think. So what we have is, we are collecting, or you would like to have your data sets. You would like to then have your methods, which we have, I've listed out over here, the methods that we work on in our laboratory, and you'd like to move them down into therapeutic interventions. Now, in the years that are coming up, maybe in the next year or two, what we need to overlay on top of the lab is the patient activity and habitual activity that you get from environmental conditions. I don't know what they are, the ones that we measure or can measure already, I've listed out over here, but there could be other things. And here's an example. For instance, is there an exciting enough application to do an image analysis from simply a patient or a prospective patient taking a photograph of what they eat? And you can break up the food into its chemical components and generate it as part of a database. So see, these are novel ways of what we are dealing with in bioinformatics today. Now, they're not traditionally driven by the biology. They are sometimes driven by the information technology aspects that come in. Yeah, so this is, I should have moved to this slide when I was talking about it. It's the same thing that I've done. This is my current group. They are haphazardly placed, so let me get down straight away into what we are doing, focused on essentially. As I mentioned, a large amount of the interest now is in molecular dynamics and structural bioinformatics. Now, this is not driven by the historical aspect of skill sets over here. It's driven by the fact that over the last 5 years, we have got very, very good traction using gaming machines with NVIDIA GPUs in large systems, equilibrium dynamics, and protein unfolding studies. So we're now moving towards the same machines can be used for deep learning with molecular dynamics, so that you can do fold prediction and residue prediction, which we basically have, which is a combination of the movement from that hidden Markov model. And eventually you might be able to think in terms of protein design. It's not so much therapeutic, but it could come in using AI. There's also a parallel project on fragment-based drug discovery, which requires AI and ligand joining methods and artificial intelligence in synthesis. Now, one of the problems with chemoinformatics is that we design molecules which an organic chemist will not be able to synthesize. And this is one of the historical issues that bioinformaticians probably missed most of their organic chemistry classes. And that's why we are not translating it. So if we can put in organic synthesis methods right into the fragment joining, then you leave out a large number of molecules which are unsynthesizable. So these are problems in AI which are coming up at the level of structural bioinformatics. Omics, which is anyway now globally a hot standard and is becoming now a little thing. There are pipelines. There is annotation, which is my personal interest, but no one younger than 30 is interested in it. But it's actually, to me, a very important exercise. So that are the standard applications of AI. There's clustering, biomarker prediction. Now, the use of clustering methods, fast and on large data sets, subsampling, etc., are the interesting aspects. There's a genome-wide association studies to variant annotations and to phenotype correlation using pathways. There's also pathway annotation. This was an aspect that I presented. We provision systems using workflows. The standard platforms that are used are the machine learning toolkit Carrot from R, what is now commonly used, scikit-learn and Python, and the Keras TensorFlow for deep learning. In text mining, these are some of the projects. And essentially, we are trying to look at building these platforms and applying them onto the previous methods that we listed earlier. These are the students, Naveen and Nitin, who are working on these areas. Okay, I end at this stage. Thank you. And